So, since we're done identifying the important concepts that we need to know before we proceed with cell division, this time let's talk about cell division. Now, when we say cell division, it can actually be classified as mitosis or meiosis depending on the type of cell. So, for mitosis, it's a type of cell division that results in the formation of two new daughter cells, each having the same number and kind of chromosome as the parent cell. So, for example, you started with four chromosomes. Okay, that's the diploid. You'll end with having two cells with four chromosomes each. Okay? So, ayun. And then, for cell division, specifically for mitosis, the type of cells that went through this type of cell division are your body cells or your somatic cells. So, for meiosis, yun yung para sa mga sex cells like gametes or your sperm cell and your egg cell. Okay, so your mitosis is actually divided into different parts. We have prophase. So, prophase, okay, these are the characteristics of prophase. Isa-isahin natin. Okay, so for prophase, yeah, chromosomes condense and becomes visible. So, if for example, this is your cell, okay, your chromosomes are already here. So, let's start with the diploid. O, kunyari, ang diploid natin or yung starting number of chromosomes natin ay 2. O, wag masyado madami so that it would be very easy. Okay, so if you have 2 chromosomes, okay, sorry for my drawing. Anyway, so if you have 2 chromosomes there, yeah, that's it. So, chromosomes condense and becomes visible. And then, you have now the spindle fibers emerge from the centrosomes or centriole. Now, what's the difference between centrosomes or centrioles? Centrosomes are made up of 2 centrioles. So, you have your centrioles here, 2 centrioles. Yay. Again, sorry for my drawing. So, this is how you draw a centriole. Okay, and then they're actually your source of spindle fibers. Now, for the spindle fibers, what we're going to do is we just connect them to the chromosomes or to the, specifically to the kinetochore of the centromere. Now, later we'll try to identify the difference between kinetochore and centromere. And then, your nucleoli and your nuclear membrane starts to disappear. So, as a representation of number 3, okay, we draw the nucleus like this. Since we draw them using broken lines because at this part, they are already disappearing. Okay, and at the end of prophase, they'll be gone. Okay, so that's it. For prophase with 2n is equal to 2 chromosome. Okay, now, next. Okay, let's try to identify the difference between centromere and kinetochore. Now, when we say kinetochore, these are made up of microtubules or proteins at the middle of your chromosome. So this is your chromosome, right? This whole part. Now your kinetochore are the fibers here. Okay, and then yung center, ano naman yung center? Yeah, that's the kinetochore. So as you can see, there's something like whiskers. Now those are your kinetochore microtubules where your spindle fibers attached to. Okay, and then pag sinabi naman nating centromere is simply a region. Okay, don't be confused. Okay, kinetochore is the structure where your spindle fibers attach, and your centromere is simply the region where you can find your kinetochore. So, yun lang yun. Okay, now let's move on to metaphase. Now, one of the famous characteristics of metaphase is that your chromosomes are lined up at the metaphase plate. Okay, so going back to our drawing a while ago. So, ano pa rin siya? 2n is equal to. So, this is the continuation. So, you have your spindle fibers. Okay. And then, you have 2 chromosomes. So, your 2 chromosomes should be aligned at the middle. Okay. And then, of course, you have your spindle fibers connected to your centrioles or centrosome. Ayan. So, that's it. For metaphase, this is how it looks like. And then, after metaphase, we have anaphase. Now, for anaphase, spindle fibers shorten so that chromatids migrate to the different poles. So, this is the time wherein your sister chromatids will be separated from each other. So, medyo elongated na yung cell on this part. And then, of course, you have again your centrioles. And then, same given for all, okay? So, it looks like this one. So, as you can see, they are already separated and they are starting to migrate from the different poles so that at the end, you'll have the same number of chromosomes. Okay? So, that's the characteristic of anaphase. The spindle fibers shorten so that they'll be able to separate and pull the sister chromatids at the end poles. Okay, and then of course we have telophase. Now for your telophase, okay, this is a bit, or there are too many characteristics or events taking place in telophase. So, first, chromatids arrive at the opposite poles and begin to uncoil. And this is the end of karyokinesis. Now, what do we mean when we say karyokinesis? The word karyo actually means nucleus, and then kinesis means separation or formation of two. So, meaning to say, when we say karyokinesis, it's simply the division of the nucleus. Next. Nuclear envelope surrounds each set of chromosome and is present again. So, during prophase, one of the events there is that the chromosomes start to disintegrate. Diba? Kaya nga, we represented it using broken lines. Now, during telophase, this is the time wherein the nucleus will form again. Okay? And then, aside from that, the nucleolus start to condense again and are present on the two newly formed nucleus. So, at the end of telophase, expect that you'll have two nucleus, and inside of which you'll have one nucleolus for each. And of course, since the spindle fibers are not needed anymore, since they've already done their duty, they will be gone. They will start to disappear. And another important feature of telophase is the formation of the cleavage furrow. Okay, so how does telophase looks like? So, again, 2n is equal to 2. Okay, so this is how it looks like. I'm really sorry for my drawing, pero wala na tayong choice. Okay. So, you'll have here the nucleolus. The nucleolus are represented by the dots. And then, of course, we have the two sister chromatid for each. And then, we have the centrioles outside na. Okay, because we already have the nucleus. Okay, so this is your nucleus. And then this is your nucleolus. And of course, we have the centrioles. Okay, and then this one here, this is the cleavage furrow. Ayan. So, at the end of telophase, you're already done with karyokinesis. So, meaning to say, you already have two nucleus. And then, each one contains sister chromatids. And of course, you already have your nucleolus. So, yeah, that's very easy. And then, so we're not yet done. So before, I think in high school, only PMAT was mentioned, but this time we'll try to include cytokinesis because this is the most important part. Now, what are the characteristics of your cytokinesis? The total separation of the cytoplasm. Okay, and of course, it's characterized by the formation of two daughter cells. So, during telophase, ano yung nag-separate? Only the nucleus. But this time, okay, we'll be separating the cytoplasm. Okay, so at the end of the process, you'll have two newly formed cells. So you have the chromatin, and of course the nucleolus. Chromatid pala. Chromatid, and then your nucleolus. Okay. Now, question. What is the state, in terms of the number of chromosomes, what is the state of the newly formed cell? Is it haploid or diploid? Okay. Actually, you might be wondering, because kalahati na lang siya, you'll refer to it as haploid. But the thing is that they are actually diploid. Okay. Remember that during interphase, kaya naging ganun yung itsura nung chromosome, or the reason why the chromosomes in prophase looks like butterfly is because of interphase. They replicated. But, okay, at the end, it's still diploid. Okay, because you have two. One, two, okay, which is like, yeah, similar to our equation, and then you'll have 1, 2. So that's how you count it for mitosis. Okay. And now, another important question that we have to answer. What do you think will happen to the cell if it divides continuously? Well, obviously, I'm gonna answer my own question. Okay. So, actually, it's not good if your cells are actively dividing, and it's also not good if they are not actively dividing. So, dapat sakto lang. Now, what am I trying to tell you? So, when cells divide faster than they should, they may develop into cancerous growth. So, one of the reasons why there are checkpoints, okay, in the cell cycle is actually to avoid the abnormal cell division or too much cell division, because what will happen, you're you'll you'll experience cancer. Okay, so ayun. Hindi siya maganda. Pero hindi rin maganda if cells divide too slowly, because your healing, or the healing of your body, would be compromised, or it will take too much time. Okay, when cells divide too slowly, wounds take a while to heal, and damaged tissues are not repaired soon enough. When cells cease to divide, as the nerve cells in the brain, memory loss results. So let's say, for example, you're wounded and you are not, or your cells are not actively dividing, then most likely your cells or your skin, the wounded part, will take a long time to heal. Kaya hindi siya maganda. Okay, now for the activity 1, okay, kindly draw a cell under the following stages of cell division. So this is like a practice. So I will be giving the answers on the next video. But for now, answer this one. And for activity 2, we also have this one. Yeah, solve for the total number of cells given the following situation. So I will explain the answers on the next video.